I'm excited to be here today to talk to you about my work, determinants of base editing outcomes from target library analysis and machine learning. But before I start, I wanted to say that MIA has been really formative for me during my PhD. I've been attending since fall of 2015. As a computational biology PhD student, the sort of intersection of the computational work and biology work that's featured at MIA has been a great learning experience, during my early years of my PhD and even till now. So it's kind of an honor and a little surreal to see myself on the other side of the podium, I suppose, though certainly not in the circumstances that I initially expected. But thanks so much for having me. So, this talk is going to be about genome editing. And so in a timely manner, I have to mention, of course, that genome editing has been recognized in 2020, Nobel Prize in Chemistry. And, just to briefly read this out loud, CRISPR-Cas9 genetic scissors, using these, researchers can change the DNA of animals, plants, and microorganisms with extremely high precision. This technology has had a revolutionary impact on life sciences, is contributing to new cancer therapies, and may make the dream of curing inherited diseases come true. So CRISPR really has been a revolution in genome editing. So for this talk, I first wanted to give a brief, definitely not comprehensive introduction to genome editing for a general audience, and then motivate a couple key problems in the field, and then discuss some of our work in trying to address some of those problems. So, CRISPR has a substantial therapeutic potential. Here's a example table of a bunch of clinical trials that are currently ongoing using CRISPR technology. This is in fact just for cancer immunology. So, one subset of medicine, and CRISPR has already had fairly large impact. And I also wanted to mention, just a couple of days ago, actually, there has been quite exciting news from several biotechs at this recent hematology meeting on advances in therapeutics for sickle cell disease and beta thalassemia using CRISPR, which has been pretty exciting. So how does CRISPR work? Before that, I wanted to briefly discuss genome editing before CRISPR, which, according to Fyodor Ornov, BC before CRISPR is one way to think about that. So before CRISPR, two strategies were prominent. So there were zinc finger nucleases and there were tau effector nucleases. And the goal of both of these strategies and CRISPR altogether is, the goal is to use a protein to bind DNA in a programmable fashion. So we would like to program the protein to bind to a specific region of DNA in our genome. And then once it's there, we'd like it to do some sort of editing activity. So before CRISPR, we primarily thought about editing activity as, first you would induce a double strand break. So what zinc finger nucleases look like here, just turn on my laser pointer, is you have an array of zinc fingers that each recognize 3 nucleotides in your DNA using a 7 amino acid variable region. And you would perhaps couple 2 zinc finger arrays that are tethered to a FOC1 nuclease. Each FOC1 nuclease is going to cut 1 strand of DNA. So when you have both arrays binding with 2 FOC1 nucleases, you can induce double strand break, and that can lead to downstream genome editing. However, zinc finger nucleases suffer from a couple of drawbacks. First, they have a fairly non-trivial mapping from amino acid sequence to nucleotide specificity, this 3 nucleotide to 7 amino acid mapping. And second, they also have non-trivial interactions between neighboring zinc fingers. So there was not a strict statistical independence that would allow you to design these zinc finger arrays easily. Your design decisions for one finger could impact the activity of a neighboring finger. Nevertheless, zinc finger nucleases were, revolutionary in their time and deserve to be mentioned sort of as a historical note today. So a bit more recently, tau effector nucleases were a different strategy for programmable DNA binding protein. They work also by a protein DNA recognition. In this case, it's a 2 amino acid to 1 nucleotide mapping, which has been solved. So you can see in this example figure, there's just 4 different combinations of these 2 amino acids that will each individually recognize a single nucleotide. So you array these together and they will recognize a sequence of nucleotides. If you array enough of them together, then you have some level of specificity in the genome, and you can precisely bind the tau effector to your target region, and then you would have a FOC1 nuclease domain again to induce a double-strand break. So in this case, tau effector nucleases were a substantial upgrade compared to zinc finger nucleases. However, in the end, they were not quite as simple and effective as CRISPR, which has really taken the field by storm. So the very basics of CRISPR-Cas9 specifically is instead of a protein DNA recognition mechanism, it uses an RNA DNA recognition mechanism where 20 nucleotides of RNA are used to recognize 20 nucleotides of DNA. So this makes it much more simple and much more easy to design. You just find whatever 20 nucleotides of DNA you'd like to match in the genome, and you can design an RNA sequence that will bind directly to that. What's notable about CRISPR and why I think it really has the most important attribute about it is that it seems like first-time experiments often just work. It's really simple, it's really efficient, and there's a lot of stories from experimentalist biologists that describe whenever there's a new technology in general, you don't really expect it to work the first time when you're trying to develop it. But with CRISPR, it seems like it really did just work. So in practice, you would design this 20 nucleotide RNA sequence, which is part of your single guide RNA or sgRNA. You would put that on the 5' end of your kind of constant part of your sgRNA, which contains a couple hairpin loops. And Cas9 as a protein is going to complex with your single guide RNA, search the genome, and then open up this R loop, this single strand kind of DNA bubble where your RNA, single guide RNA is going to bind to your DNA target sequence and leave this other single strand DNA strand open in this bubble. And the native activity of Cas9 is that it will cleave both strands and induce a double strand break. Another important feature about Cas9 is that it requires binding of this recognition of this 20 nucleotide region called the protospacer in your single guide RNA. And it also requires what's called a protospacer adjacent motif. So in the case of Cas9, that is NGG. So it will recognize 20 nucleotides plus 3 nucleotides NGG in order to be specific. So I think a different and more powerful way to think about CRISPR in general is simply as an RNA-programmable DNA-binding protein. So the CRISPR natively will induce a double-strand break. In my opinion, some of its best uses really treat it just as an RNA-programmable DNA-binding protein again. So here, if we consider kind of standard CRISPR-Cas9 editing, these blue triangles would indicate nickase marks. So you're nicking both strands of DNA, and that induces a double-strand break. Instead, some more recent genome editing technologies, including base editing, instead we remove one of the nicking activities. So we only nick one strand of DNA, and we tether other proteins and other components to the Cas9. So basically using Cas9 as a platform, it's a homing platform to deliver these other components to this target region in your DNA with high specificity. So in the case of base editors, we've tethered a deaminase, which is going to deaminate specific nucleotides alongside a component called UGI, which I'll briefly discuss later. In the case of more recent prime editors, instead what we've done is tethered a reverse transcriptase to the Cas9 nickase. Again, we're only nicking one strand of the DNA. And we, instead of using a vanilla single guide RNA, they have designed a more unique what's called a PEG RNA or prime editing guide RNA. So by thinking a bit more creatively and a bit more abstractly about CRISPR as simply an RNA programmable DNA binding protein, these are just some of the things that you can do. And of course, this barely scratches the surface of what's possible with CRISPR. So genome editing with CRISPR-Cas9. So sort of in the vanilla standard approach, back in the days where we're primarily thinking about causing double-strand breaks. What happens after a double-strand break in order to lead to your desired genome editing outcomes are a series of DNA repair mechanisms. So here after your double-strand break or DSB, there's a variety of different possible repair mechanisms that could occur, including end joining mechanisms, and also homology-directed repair, or HDR. So in the context of HDR, we would like to spike in some sort of donor or template DNA And this is sort of exploiting the fact that a double-strand break is kind of a catastrophic, kind of damage to the genome. And if the cell is not able to repair a double-strand break effectively, upon division, it will die. So it's really serious news. The cell needs to repair this effectively and quickly. So in homology-directed repair, where the cell will natively try to use the meiotic sister chromatid sequence to repair a double-strand break in a specific chromosome. But we trick the cell by providing donor DNA that looks similar to the region that has been cut. And we hope that the cell will use that instead of the sister chromatid to repair the lesion. And this can result in targeted integration of a designed sequence into the genome. So in principle, the homology-directed repair strategy is capable of installing many different kinds of edits, including small insertions, small deletions, point mutations, combinations of these, and etc., which in principle allows for a very flexible installation of any desired edit that you would like. However, it turns out that homology-directed repair is often inefficient, and it mostly varies by cell type. So homology-directed repair, its mechanism requires cell division, which in many kind of adult cell types no longer happens very frequently or at all. So in many cell types, it turns out that end joining repair mechanisms are usually much more efficient. However, from a genome editing perspective, they are often quite undesired. So these end joining mechanisms, such as non-homologous end joining or alternative non-homologous end joining, typically result in indels or insertions or deletions at your target loci, and they're generally uncontrolled. So, around the initial advent of CRISPR-Cas9 genome editing, this was the main strategy, is you would hope for homology-directed repair to install your desired edit, and you want to minimize your uncontrolled indel byproducts. For these reasons, the strategy of homology-directed repair and associated end joining products has been called genome vandalism by George Church. And by many people in the field, indels were thought to be unpredictable and generally not useful beyond gene disruption in order to cause frame shifts. You're not able to control the insertions or deletions that occur. They're very heterogeneous, and they cannot, for example, encode point mutations. So I briefly wanted to discuss the mechanisms also for base editing and prime editing. So base editing, we've tethered a cytosine deaminase to your Cas9, and this is taking advantage of the fact that this is a single-strand DNA-specific deaminase, and it can target cytosines on the other side of your R loop, which would deaminate the C to a U. Typical cellular DNA repair mechanisms are going to try to extract the U, which does not belong in DNA. The protein responsible is uracil DNA glycosylase. This would form an abasic site, which upon base excision repair would result in a correct repair of your original C. However, for base editing, we would like to convert the C to a T, which T is read as U or U is read as T. So what base editing does is it nicks the non-edited strand here with a G, which tricks the cell into thinking that G is the problematic incorrect base, which could lead to repair of the mismatch into a U-A pair. And upon DNA replication or repair, this can lead to a permanent installation of a C to T edit on the double-strand DNA. So one of the key insights for the base editing mechanism is that by avoiding a double-strand break, base editing is able to avoid indels from end joining DNA repair. So some key attributes about base editing is that an adenine to inosine deaminase was later evolved, which does not occur naturally, and that together is able to enable all transition conversions. There have been some recent attempts at transversion base editors earlier this year, and generally base editors are able to efficiently edit single nucleotides in a 5 to 10 nucleotide window with high purity. So these transitions are, for example, between A and G or between C and T. And you can see that, for example, purines have 2 rings, pyrimidines have 1 ring. So it makes sense that chemically we would be able to easily convert from A to G or from C to T, but it could be more difficult to achieve transversions. So the ability to install transition point mutations is pretty important and impactful for therapeutic reasons. because about 2/3 of pathogenic SNPs can be correctable by transition editing. However, 1/3 can only be corrected by transversion editing. So this is the prime editing mechanism. I'm going to briefly gloss over this for time reasons. The main takeaways for prime editing is that if we look at the distribution of all pathogenic genetic variants, the previous plot I showed on the previous page just considered point mutations, but that is about just half of all known pathogenic genetic variants. And the other half of the pie chart are deletions, duplications, copy number, loss and gain insertions of different kinds, which are not amenable to efficient correction by base editing. So prime editing is able to use a very clever and creative mechanism in order to flexibly install not just point mutations, but also short insertions and deletions while avoiding a double-strand break in indel byproducts by avoiding inducing a double-strand break in the first place. So a brief comparison of prime editing and base editing is that for edits that are possible by both mechanisms, base editing generally is more efficient and has fewer indels. So prime editing is substantially more flexible, but for now remains a bit less efficient than base editing. So zooming out a bit at some, in my opinion, important questions about genome editing. For example, we would like to know how specific is CRISPR genome editing? And this has received a lot of attention, research attention, since 2012. I'd just like to point out that computationally, this is a pretty tricky problem, even though it has attracted a lot of attention. Observed off-targets are fairly sparse relative to the entire size of the human genome, though there has been some interesting work on kinetic models of binding and dissociation for this. Another important problem that we'd like to ask is how efficient is CRISPR genome editing in the first place? Can we predict this from sequence alone? And there has been some work on machine learning, targeted towards this question. It has also received a fair amount of attention. Initially, models were typically built on small data sets with fairly indirect measurements of efficiency. However, more recently, there have been, we've been able to expand data set sizes by a variety of groups and also obtain more direct measurements of efficiency. So these are off-target activity. This is one question of on-target activity is efficiency. Another question of on-target activity are what are the actual genetic outcomes of CRISPR editing? And to some surprise, this wasn't really addressed until 2018 by us and other groups. So briefly, coming back to DNA repair outcomes following Cas9 cleavage, here we have this double-strand break. And I mentioned before that these uncontrolled indels from end-joining products are typically very efficient, but usually undesired, and were called genome vandalism. So in a long story short, on one of our previous papers, which was published in Nature in 2018, we, in collaboration with experimentalists, we measured genotypes after Cas9 cleavage at thousands of design target sites in human and mouse genomes. We presented a multitask deep model designed based on DNA repair mechanisms that was able to accurately predict heterogeneous insertions and deletions. And we used this trained model to discover a new genome editing strategy for efficiently correcting a class of pathogenic mutations called microduplications. So, long story short, could genome vandalism actually be interpreted as genome art? So, understanding the actual genetic outcomes of CRISPR editing, I think, is important for therapeutic and also research applications. So that is kind of the brief overview and not comprehensive overview of genome editing. Now I want to talk about characterizing base editing outcomes using largely a similar approach to what we did for Cas9 outcomes. So this is some example data of what you might see if you do base editing at a single loci and kind of do deep sequencing and analyze the results. So in particular, this is comparing base editor 3 in this top section to Cas9 and homology-directed repair in this bottom section. And the goal here is to change this arginine to a cysteine. And with base editing, we can see that they're able to achieve quite efficient conversion of C to T nucleotides roughly targeting these three cytosines all together. Since base editing targets a small window, the R loop and the single-strand DNA exposed by Cas9 binding has a size of maybe 20 nucleotides or so, and the single-strand deaminase is able to deaminate all cytosines within that small 20 nucleotide window, though the actual range of base editing is a little smaller. So in practice, we see that these three cytosines are edited. We also note that this cytosine in this neighboring amino acid, leucine, was also edited at about 50% frequency. Half of the C's were converted to a T. However, in sort of by luck or by design, that did not affect the amino acid sequence. So this leucine remained a leucine, and this mutation was silent. So in practice, this specific application is correcting an ApoE4 a mutation allele in mouse astrocytes. So this is quite therapeutically relevant. And this is just from one of the base editing papers. So they compared to homology-directed repair, where there was essentially 0% efficiency in this particular cell type. So this sort of motivates, again, base editing can be very efficient at point mutations, and homology-directed repair can struggle with efficiency. So with this homology-directed repair approach, they also observed very high indel frequencies of about 26%, while for base editing, they had about 5% indels compared to about 50% on target editing. So there's a variety of questions that arise. Would it be possible to foresee these sorts of base editing outcomes in advance just from sequence alone, for example? And are there sequence preferences for these deaminases that might be able to discover or be able to tell us about what sorts of outcomes we would be able to expect? So in general, studies that present new base editors measure activity at a few dozen target sites at most, and they might typically report estimated editing window width and position, and also estimated sequence preferences from deaminases. So, sort of this catalog of about a dozen or so different cytosine base editors, some of their editing windows here are listed maybe from, position 4 to 6, so about 3 nucleotides, or positions 4 to 8, or about 5 nucleotides or so. Some of them are wider, from position 1 to 10, or about 10 nucleotides targeted. And some of these sequence preferences that are known from the literature, just from characterizing them at a few dozen target sites, are relatively simple motifs where they might prefer TC since it's presented in green, and they might strongly prefer GC since it's presented in red, while other base editors might have no real sequence preference and be described as NC. So if you were to design a base editing experiment, you would be looking at sort of these editing window sizes and these sequence preferences to try to make a decision. So I would also note that many distinct base editors have been developed. In fact, I would say that this is relatively easy. Of course, nothing in research is really easy. But to this day, there are far more base editors than CRISPR family members, in fact. So one strategy, for example, to design a new base editor is to do circular permutation of the deaminase. So if you just take your deaminase and you change where the N terminus is and where the C terminus is to start at some middle amino acid, by doing this sort of circular permutation, you can actually change the deaminase editing activity substantially. So, to date, there have been dozens of base editors with distinct editing windows, distinct sequence preferences, distinct editing activities in general developed. And it's sort of an explosion in this landscape. So these myriad base editing strategies that are available, in my opinion, they motivate data-driven design principles, which have been largely lacking to date in the field. So in order to achieve a goal phenotype, we might consider multiple possible genomic edits, for example, that edit the amino acid sequence in different ways. We might consider, for each of these edits, multiple base editors, again, among the dozens that have been developed to date. And for each of these editors, there might be multiple guide RNAs that would be able to install these desired genomic edits with varying efficiency and also varying bystander precision, since you would be able to, in some cases, edit neighboring cytosines. And in this previous example I described, it could be a silent amino acid mutation, but in other cases, it might not be a silent amino acid mutation. It could be very deleterious. So if you consider base editing efficiency and the bystander precision together, that should give you the total frequency of the desirable base editing outcomes in your sequence to reads. And you'd like to choose among all of these myriad base editing strategies, and it would be nice if you could do so in a quantitative kind of rational manner. However, in practice, users have needed to try multiple base editors and guide RNAs in order to optimize their experimental designs.
Max, why do you say goal phenotype as opposed to goal genotype here?
So, for example, if you wanted to knock out a gene, you could introduce a stop codon at a variety of places, and some of them may be more easy to do by base editing, some of them might be more difficult.
Right.
So yeah, the genotype to phenotype link is a whole, maybe a core research problem for the next 100 years. If we could really crack that, that would be great. But as it stands,
it's sort of any of the genotypes that realize the desired phenotype here. Is that?
Yes.
Okay.
Yeah. Yeah, and I would note that in practice, genotype to phenotype mapping is one of the hardest problems, in my opinion, for designing good genome editing strategies.
Sure. So do you mostly have in mind examples like either a knockout with a premature stop or a frame shift, or, for example, a single amino acid substitution where there might be a couple of synonymous options, or?
For our work, we primarily focus on, since we're using base editing, the strength is single nucleotide conversion. So we're primarily focusing on fixing single nucleotide polymorphisms directly in the genome, or correcting amino acid mutations using base editing. Yeah.
So in that case, there is really just one or a very small number of gold genotypes that you can identify.
Right, yeah. So if you're looking at amino acid sequence, you would like to convert a mutated amino acid sequence back to wild type, there could be a handful
right.
of possibilities.
Right, okay, thanks.
Yeah. So this is an example, designed decision tree diagram from a recent review on genome editing on how you might design a base editing experiment. Of course, this bottom section is about off-targets, which is less relevant to what I'd like to focus on today. However, this could be a little bit complicated, and it would be nice if you could do so in a kind of a quantitative way. For example, also some of these boxes ask, are bystander edits acceptable, or are there silent or conservative mutations, or are they less likely to be edited? This is also something that really depends on the editing window size and also sequence preferences of various base editors. And it can be difficult to walk through this decision tree in a completely objective manner without inserting some of your own knowledge or lack of knowledge into this decision problem. So our approach is to measure base editing activity at 10,000 or tens of thousands through the course of the work, single guide RNA target pairs. So we characterized 8 base editors with 2 replicates in a variety of mammalian cell types using both adenine and cytosine base editors. We designed a minimally biased characterization library where a fair number of our target pairs had either an A or C at protospacer position 6, which is where base editing activity is typically the highest, surrounded by all possible combinations of ends around this central substrate nucleotide. We also designed a fair number of target pairs that varied all possible nucleotides from protospacer position 1 to 11, which is broadly speaking the base editing window for many of these base editors, centered at 6, spanning from about 1 to 11, with all possible nucleotides in positions 1 to 11. So we call this the minimally biased characterization library, or comprehensive context library. And what we're able to do is synthesize oligos containing these designed guide RNA target pairs, introduce them into cassettes, which are integrated into mammalian cells, alongside a base editor cassette. And these sequences are integrated into the genome. The base editor is expressed, and there's base editing activity. Following base editing activity, we do deep sequencing high throughput sequencing in order to read out a variety of the base editing outcomes at these designed targets in the context of the native genome. So this work, I should mention, is all done in close collaboration with Monda Arbab, who's a postdoc in the Lu Lab and co-first author on this work, and I couldn't have done all of this without her. So the high-level picture, and taking a step back briefly, and the goals is, I would like to note that base editing had not been characterized at this scale before. And in fact, there was not really a precise or quantitative definition of base editing activity before. There was no widely accepted communal standards on separating signal from noise for this kind of data. So of course, this is the norm when you're working with scientific data at sort of the frontier, when you're generating a data set that of a certain scale and size that people have not done before. So this, introduced some computational challenges that had to be overcome. And broadly speaking, our motivating goals, some of them at this stage, was to achieve a minimally biased characterization of on-target base editing activity at unprecedented resolution, and to understand the sequence preferences that drove efficiency and also bystander editing patterns. So one thing to note in our experimental setup is that we perform 2 steps of antibiotic selection, where each selection step kills about 90 or 99% of cells. And these selection steps help ensure, this first one helps ensure that all of our surviving cells have a library cassette. And the second antibiotic selection step ensures that all of our cells have both the library cassette and a base editing cassette, in order so that all of our cells can undergo base editing. So because these selections are a bottlenecking event, we see that rare background mutations that can occur before any base editing activity, for example, in our library target sequences, because they undergo bottlenecking events, they basically undergo this sampling procedure where they're drawn from a low N, high variance normalized binomial for each library member, and this can cause inflation of background mutations. And since background mutations are often point mutations, this posed a problem in order to be able to sensitively look at real base editing activity, which is typically, again, introduces point mutations. So we took a variety of typical standard statistical approaches in order to process this data, including quality filtering, removing PCR recombination products, sequence alignment, adjusting for luminous sequencing errors, adjusting treated conditions with untreated control data, et cetera. So in order to try to minimize bias as much as possible, we chose to try to investigate all possible single nucleotide mutations in this 56 nucleotide region around the protospacer, which we designed. So here, this is all A to all other possible nucleotides. This is C to all other nucleotides, G and T to all other nucleotides. And we wanted to say, let's pretend we know nothing about base editing in order to be as minimally biased as possible. What is real signal in the data? So it turns out that these selection steps essentially cause batch effects. So if you're able to perform many batches, then you're able to correct for them using one-way ANOVA in order to correct for these batch effects. These point mutations can be quite inflated and quite high frequency in some cases. However, if we have enough batches, we're able to pick up signal that reliably appears across batches. And after doing this batch effect correction, we're able to see a very kind of pretty, kind of smooth window bell curve reflecting what seems to be real base editing activity, in this case, A to G editing. So we wanted to be able to use the scale of our data at tens of thousands of target sites to be very sensitive as well about what on-target base editing activity might look like. So we also used an approach called robust rank aggregation in order to try to identify events that occur consistently above background frequencies across treatment replicates. So by using this approach with our large library, our deep sequencing depth, and multiple replicates, we were able to confidently identify events that were replicable to frequencies as low as 0.01%. This is sort of what robust rank aggregation looks like across multiple replicates. we want to see, even if their log probability is extremely low, like -5 or something like that, they can be considered significant in orange if they are consistent across replicates. So the results of these procedures was we were able to identify these sorts of plots for each of our base editors. So this is for one base editor. These are 4 base editors presented on this slide. And we're able to consider all possible single nucleotide mutations in kind of a minimally biased and sensitive manner. So there were some interesting observations that came out of this attempt towards sensitivity and minimizing bias. For example, we could see that for a base editor, adenine base editor, its core, most frequent activity as expected is editing A to G in the central red column. However, we can see that there seems to be sort of smooth but very low frequency bell curve window around C to T and also C to G point mutations that is also centered around protospacers positions 5 to 7, which is where the primary A to G editing activity occurs. So these numbers are normalized to the maximum of frequencies observed in our data. So this is 1 part out of 100. For every A to G edit, there's less than 1% C to T or C to G edit. So these frequencies are quite low and would not be picked up if we were not as sensitive. So we're able to observe some other editing activity as well. So for these cytosine base editors, the central editing activity in red is C to T editing. However, we can see pretty substantial C to G editing by and C to A editing by some of our cytosine base editors. So the canonical BE4 base editor 4 has about 5 parts in 100 for C to G editing, while EA3A, another cytosine base editor, has up to 10 parts in 100 for C to G and C to A editing. What's also interesting here is, again, this very rare kind of activity, which appears to be a G to A editing around protospacer positions above 0, 5' of 0, which can be considered, it's quite noisy, but about 1 part in 100 for every on-target C to T edit. So there were similar stories for many of the other base editors. For this other base editor, BE4CP, the circular permutant, this G to A editing 5' of the protospacer is a bit more relatively frequent. Another observation we were able to do here is to quantitate the base editing windows with much higher, finer resolution than previously. Using data at just a few dozen target sites, we're able to build on that and develop these really kind of clean and nice, very precise editing window widths that like smoothly decay down from 100 towards 0. So we wanted to investigate this sort of aberrant rare activity, and we built some motifs in order to, and we found that they suggested mechanisms that could be underlying these rare outcomes. So
now it's more.
No, it's more.
So in particular here, we investigated A-base editing and the rare cytosine editing by these A-base editors. So here is the base editing efficiency motif for A-base editors at their canonical A substrate. And we can see that it prefers this sort of TA motif, which has been reported by other people, but we're able to see contributions from farther away than just one nucleotide from the A. And if we look at the motif governing presence or absence of this rare cytosine editing activity by the adenine base editor, we can see that the motif is very similar. It's just centered around a C instead. So this is what the activity that was happening, less than 1% relative frequency to the A base editing. So this similarity of motifs suggests a mechanism where the adenine base editor deaminases can directly deaminate cytosine. And this was a novel finding at the time and has actually been corroborated by other groups. We also looked at rare guanine editing by these cytosine base editors that was occurring 5' of the protospacer. So what's interesting here is that the motifs for base editing efficiency at essential C, which may prefer TC as reported by previous groups with other contributions from farther away nucleotides, seems to be reverse complemented when we look at the motifs governing presence or absence of rare guanine editing by CBs. So these motifs suggest that G is being edited as opposite strand cytosine editing. And again, these frequencies occur very rarely, but we were able to sensitively pick them up through our careful analysis. So we also investigated rare indels. They're quite rare. So if you compare the on-target base editing activity to the indels, you get ratios about 40-1 or greater than 100-1 for a variety of base editors. But I think I will quickly skip through this part. For now, I just wanted to quickly highlight this plot where we were able to very sensitively look at where indels occur, the position of indels. And because they're so rare, this has been very difficult to do before. But with the scale of our data set, we were able to pull out plots that look like this, that implicate indels that are in some cases being caused by single-strand DNA nicks from the formation of abasic sites, if you recall from the mechanism I outlined before. So next, we investigated base editing activity at disease-related SNPs. So we designed libraries of about 20,000 disease-related SNPs from ClinVar and HGMD that were correctable by base editing of A to G or C to T, so using the same library approach as before. And one observation that was pretty surprising to us is just the sheer amount of variability in bystander editing precision at these disease-related SNPs. So the previous common sense for base editing is if you have 5 Cs in your target window, then of course your editing outcomes will be less precise than if you had just 1 C in your target window. However, we observe substantial variability in the precision of outcomes, even when you controlled for the position and number of bystander substrates. So to explain that in this plot here, we've applied a mask on all the nucleotides in this range, except for these 2 Cs, which are Cs in all of these different pathogenic SNP target sequences. And the number here in blue indicates the precision of outcomes that precisely correct just the single C to a T and do not affect the other C at position 3 here. So you can see that the starred C is precisely corrected in some cases with frequency as high as 93%, and in other cases as low as 6%, even though by conventional wisdom, the primary factor determining bystander precision should be the number of Cs and the relative position of them. But even when controlling for the number and position of your bystander Cs, the substantial variability indicates that more complex sequence preferences were at play. So it's the same story here for adenine base editors. Even though we just have 2 As in this region and they're all at the same fixed position, the single nucleotide precision of correcting the starred A ranges from as high as 99% purity down to 0% purity. We also observed that so-called non-core substrate nucleotides could also substantially influence bystander precision. So in this case, what has been canonically defined as the core editing window for this base editor, we mask out target sequences such that they only have a C at the central position 6 and do not have a C anywhere else in the core editing window. However, they could have a C outside of the core editing window. And we still see substantial variability in single nucleotide correction position of this C6, which indicates that in some cases, these Cs that are outside of the core editing window, for example, in this case, there's a C all the way up to the left here and no other C except for the C6. So in some cases, this C all the way to the left can be edited more efficiently and more purely than your C6, which is at the center of your so-called editing window. So the previous thinking here was that base editing activity can be approximated well just by sort of core window size of about 5 nucleotides. But in some cases, we show that this core window can really be misleading when you look at real data. So for our modeling approaches, if we are able to model base editing efficiency and also bystander precision together, then this is able to, we can combine the predictions from both segments of the model in order to predict the frequency of any base editing outcome subset among sequenced reads. So for base editing efficiency, this is very simple. We tried deep learning approaches, but in the end, gradient boosted regression trees were the most effective in generalizing with the amount of data that we had, which was about 10,000 labeled inputs. So we used pretty standard features for the gradient boosted regression trees. We're able to predict a z-score, which can be optionally calibrated to the user's experimental setting in order to adapt to other cell types and other delivery methods, for example. And this allows us to accurately predict the fraction of sequenced reads with base editing activity. So the other model for bystander precision must solve a sequence to distribution of sequences prediction task. So what I mean by this is that we would like to predict from an input genomic sequence, which is just a single sequence, the frequency of the distribution of all possible editing outcomes, which is a distribution over sequences. So in this case, one possible outcome is that just a single C is edited to a T. Another outcome is that this C is added to a G, or that two C's are edited to a T. So you have, combinatorially many possible outcomes. So to solve this task, we use a deep conditional autoregressive model architecture, which uses a deep encoder on all of the substrate nucleotides in our input sequence context. And we use a deep decoder that autoregressively conditions on previous outputs from the model in order to predict the probability of any combination of outcomes. So at each step, we kind of move in an arbitrary order from left to right here. We encode the substrate nucleotide, and then we combine that with information from previous decodings in order to predict an outcome frequency among 4 possible nucleotides. We sample the editing outcome, and then we condition on that in the next step of the model moving forward in order to sample a new nucleotide. And we do this for every substrate nucleotide in the input sequence context. So I think one perspective that's interesting here is that distributional output is a much richer signal than a regression output for the efficiency model. So compared to the efficiency regression task, we have about, in some sense, 10 to 1,000 times more data per library member, which is a total of up to 10 million event frequencies. And we find that this enables deep learning to be effective and to learn to predict quite accurately. So another feature of our approach is that this autoregressive decoder directly outputs normalized probabilities, which is very nice. And it does not require us to have to enumerate all combinatorially many outcomes in order to compute the partition function. So briefly, this deep conditional autoregressive approach works by decomposing a joint distribution over any number of substrate nucleotides and what they could be edited to. So in this case, we might consider 4 nucleotides that are edited to a variety, A, C, G, or T. The joint distribution can be decomposed as a product of conditional distributions where we calculate the probability of a single new nucleotide outcome conditioned on everything that we have observed before. So this can be more succinctly written using this notation. So while the number of substrate nucleotides in a target can vary, we can use a conservative upper bound and the total editing window size to obtain a fixed input shape to a residual net, which is what we do for our decoder. So one way to understand what we're doing here or how to implement this is use a masked context. So here, everything in white is in the future, and we mask that out. Everything in blue is in the past, so we use previously sampled outputs as values here, and we'd like to predict X1. So this example diagram is in the case of an image, but you can generalize to the case of our 1D DNA sequence as well. So to predict X1, we predict a sample of single nucleotide for X1, and then you repeat the process, putting your sample into this non-masked region, and we'd like to predict the next nucleotide, and we move forward until we have completed this for all of our substrate nucleotides. So the results of our model is that, again, on this sort of masked, controlled sorry, let me drink some water.
Max, just wanted to give you a time check. It's maybe just a few minutes left.
Yep.
Great.
So on this observed variability for all of these variety of outcomes, we find that our model is able to accurately predict the single nucleotide correction precision for the single target nucleotide, which closely matches the observed variability in your precision. We show this also for the adenine base editor. And more broadly, we can extend this to all of the base editors that we see. So for some of our base editors, their precision is better than others, and our predictions are able to closely match the observations. So our model is surprisingly well calibrated and also quite accurate across our disease library, libraries containing disease-related SNPs of about 20,000 disease-related SNPs. Our correlation with between our prediction and observed is as high as 0.92 for some of these editors. So these results on held out data show that we're able to very accurately predict single nucleotide correction precision using our deep conditional autoregressive model. Moreover, while the conventional wisdom might say that at sites where you have multiple substrate nucleotides, you should expect low correction precision, and indeed the baseline is between 3 to 36% precision, we were able to experimentally demonstrate greater than 90% correction precision for about 500 pathogenic SNPs using our model. So our model is able to accurately and a priori identify these SNPs where you can achieve very high correction precision despite the presence of multiple substrate nucleotides, which, from before, from heuristic guidelines, you might just disregard those as targets. So very briefly about cytosine transversion base editing. So I motivated before that standard base editors can only do transitions and not transversions. However, 1/3 of pathogenic SNPs are transversions. We also observed previously, I noted that EA388 seems to have elevated baseline transversion rates. So we wanted to ask if we could discover the sequence determinants governing cytosine transversion base editing and see if we could use that to efficiently or purely correct transversion SNPs for the first time. So from motif analysis, we were able to discover motifs that were generally predictive of cytosine transversion base editing activity with a Pearson correlation of about 0.5 for a variety of these base editors. And they seem to prefer this sort of R-CTA motif where R is G or A. So this is just a standard linear kind of independent motif. We can also tackle the same question using our deep conditional autoregressive model. And we're able to do so, and we're able to observe that our model is pretty predictive with a correlation of 0.78 when we ask it to predict the frequency of repair to wild-type amino acid sequence and compare that to observed precisions. So we did this experiment in a new 4,000 target library, again designed using ClinVar and HGMD that contains transversion SNPs. And what's exciting is that we're able to demonstrate for about 100 transversion SNPs greater than 90% purity correction to their wild-type amino acid sequence for the first time. And this is kind of building off of our model's ability to accurately predict the sequence determinants of transversion base editors and bystander editing patterns. So in summary, we sort of did an encyclopedic kind of characterization of base editors. We provided insights into rare editing outcomes and interactions with DNA repair processes. This is more in our paper. Our model was able to accurately predict bystander editing patterns, transversion base editing, etc. We provide an interactive web app that is used by hundreds of scientists every month at CRISPR-BE-Hive.design. This work was, to my honor, featured on the cover of the July 23rd issue of Cell, actually with my pixel art that I designed. So that's kind of an incredible thing that I did not really expect in any world would happen. I want to note that this work was all done jointly in close collaboration with Manda Arbab again. We're co-first authors on this work. We have consented to either ordering, so I present myself first in this talk. The publication order has Manda first. The paper is a lengthy paper that covers way more work than I was able to discuss today, including a lot of really exciting work spearheaded by Manda on developing new base editors with improved capabilities, building off of our library characterizations. So I wanted to mention that I'm actively looking for opportunities for the future. I'm ready to defend my PhD at any time. I'm passionate about advancing the ways that computers can solve problems using machine learning and translating those advances to scientific discovery and biological impact. This is, this work and other work collectively. I want to encourage anyone that is attending NeurIPS this year virtually to attend my invited talk at the LMRL workshop on Friday. With that, yeah, I would like to acknowledge my advisors and everyone that helped out with this work. And thank you so much for your attention.